Welcome back, my dear students. So in the last class we saw how IMP, AMP, GMP etc. are synthesized, the synthesis of purine nucleotides. Now in that I said to you what is important was glutamine, carbon dioxide, aspartate, all these act as the donors of the different carbon, nitrogen or hydrogen atoms, carbon and nitrogen atoms in this purine ring. Now just for the sake of MCQs, I have redrawn this purine ring here. Let us look at what are the contributors of the different atoms in the purine ring. So the purine ring is numbered starting from here. 1, 2, 3, 4, 5, 6, 7, 8 and 9. That is how the numbering goes. So whenever in the MCQ it is asked, they will ask you which of these are contributed by what and all that. So if let us look first at glycine. The whole of glycine molecule is going to be incorporated into the purine ring. So it will contribute to C4, C5 and N7. C4, C5, N7 comes from glycine. You look at C6. C6 comes from carbon dioxide. Let us first talk about the different amino acids. The different amino acids include glycine, glutamine and aspartate. Now glycine contributes C4, C5, N7. Glutamine contributes N3, N9. N3, N9 by glutamine. N1 comes from aspartate. So the three amino acids and their contribution. So C4, C5, N7 glycine. So one nitrogen. The other two nitrogens N3, N9 come from glutamine. N1. So totally 1, 2, 3, 4 nitrogens are there. One nitrogen comes from glycine. Two nitrogens comes from glutamine. One nitrogen from aspartate. So the amino acids are over, which are contributing the different nitrogen groups. Now let us look at the carbon contributors. Carbon, we have seen glycine contributes to C4, C5. Other than that, carbon comes from carbon dioxide. The other carbon donor is N10 formyl tetrahydrofolate. So it's very easy. The N10 formyl tetrahydrofolate contributes C2 and C8 of the purine ring. So we have the 4 nitrogen atoms and the contributors of the different carbon atoms. 4 nitrogen atoms come from glycine, glutamine, aspartate. Glycine N7, N3, N9 from glutamine, N1 from aspartate. And you have got the different carbon atoms. One comes from carbon dioxide, C2 and C8 come from N10 formyl tetrahydrofolate. The remaining come from glycine itself, C4, C5, N7 from glycine. So this is how the different groups are brought together during de novo synthesis. So what is happening? Raw products are converted into a final compound, each contributing different portion of the purine ring. This is also an energy requiring process and requires ATP etc. for this to be formed. Remember, this is a purine ring, and depending on the additions of the side chain, we have got different like inosine, guanosine or adenosine. Adenine, guanine and iodine are formed because of the addition of the different groups in the purine ring. This is the contributor. MCQ point of view, you need to know about it. Now let us look at the different inhibitors of the purine synthesis. So let us look at the inhibitors of different of the de novo synthesis of purine nucleotides. One important inhibitor is PABA analogues, all the PABA analogues. Now sulfonamides are structural analogues of PABA and they competitively inhibit folic acid synthesis in bacteria. The same thing is required even in bacteria. So in the bacteria, if folic acid formation is not taking place, it will lead to effectively no synthesis of purine and de novo synthesis will stop. De novo synthesis will stop means what? It will also lead to the stopping of DNA synthesis and thereby the cell will not divide. So sulfonamides will bring about their effect on the bacteria by acting as inhibitors of tetrahydrofolic acid formation, the active form of the THFA. So they are structural analogues of PABA and competitively inhibit folic acid synthesis in bacteria. Now, purine synthesis requires DHFAS coenzyme. So, this will lead to inhibition of the de novo synthesis, inhibition of DNA, and its effect on bacteria. Now, that is when the bacteria, you want to kill those bacteria. You don't want the bacteria to divide. But there are certain times within our body when we don't want the body's cells to divide. When cancer. So in cancer also, when a cell is dividing, it is going to synthesize DNA. If it is going to synthesize DNA, it is going to use the de novo pathway to synthesize AMP, ATP, GMP, etc. So we have what are called as folic acid analogs. Now, one of the drugs that is known is methotrexate. What it does, it inhibits dihydrofolate reductase. So it prevents the conversion of dihydrofolic acid to tetrahydrofolic acid. So this conversion is inhibited, and thereby it slows the DNA replication in human cells, and hence used in cancer chemotherapy. Now there is one more, that is mycophenolic acid. Now this inhibits IMP dehydrogenase. Please remember, IMP dehydrogenase was involved in conversion of IMP to GMP with the formation of XMP as the intermediate. So this XMP formation, inosine to XMP, requires inosine dehydrogenase. This enzyme is inhibited by mycophenolic acid. This is a type of uncompetitive inhibition. So what it does, it deprives the rapidly proliferating T and B cells of nucleic acids. The T and B cell are deprived of the nucleic acid, and mycophenolic acid is used to prevent graft rejection. Especially to prevent graft rejection, we use mycophenolic acid. We'll be learning more about this in different years as you go ahead. Right now, PABA analogs, folic acid analogs, and mycophenolic acids. Now, as I said, this was all de novo synthesis of purine nucleotides. And there is one more way by which purines can be synthesized in the body, and that is the salvage pathway. So, let us look at the salvage pathway for purines. What is this? Salvage. When do you salvage something? When something is going to go beyond, you try to save it. You call it salvage. You are trying to save something which could have got degraded. You tell it, we are trying to salvage something. So you are trying to save something. You don't want to unnecessarily, the body doesn't want to utilize a lot of energy for this de novo synthesis. So it is going to salvage it. So the salvage pathway is very important in tissues like erythrocytes and polymorphonuclear leukocytes and brain. So why in this tissue? Because de novo synthesis of nucleotides is either of limited capacity or does not occur in these tissues. So for these tissues, salvage pathway forms the only route by which they can do their nucleotide synthesis that is required. So what to do? How to get about these nucleotides? They can utilize purine and pyrimidine derived from other tissues to form their own nucleotides. So that is what it does. Either the purine derived during the normal turnover of the cellular nucleic acid or the ones which are obtained from the diet. Both of these can be utilized because of the salvage pathway. These reutilization pathways are called as salvage pathways. We have a similar reutilization pathway supposed to be there for pyrimidines also. However, that is not very effective. The different reactions in the salvage pathway are all involved the reaction of the bases with PRPP. Again, we got that phosphoribosyl pyrophosphate has again come for use in the salvage pathway. So, what does it mean? It means ribose 5-phosphate is important not only for de novo synthesis but also important for salvage pathway also, because PRPP is involved. So basically it means that glucose HMP shunt pentoses ribose 5-phosphate is important not only for de novo synthesis but also for the salvage pathway also, because PRPP is required. So let us look at the two very important enzymes involved in the salvage pathway. I have just written down the reactions here, and the names of these enzymes. They look like complicated names, but abbreviations are simple and easy to use. Now the first enzyme is hypoxanthine guanine phosphoribosyl transferase. It is abbreviated as HGPRtase. What does it do? Remember, PRPP is used here. So hypoxanthine is converted to IMP and the donor is PRPP. So conversion of hypoxanthine to inosine monophosphate. Conversion of guanine. What is hypoxanthine? Hypoxanthine is a base. And what is IMP? It has formed a nucleotide. So the conversion of a base to a nucleotide by donating the phosphate group and the sugar group to the base is a part of the salvage pathway. So in the first pathway, hypoxanthine combines with phosphoribosyl pyrophosphate PRPP to form inosine monophosphate. And the same enzyme can catalyze another reaction. That is, it can salvage the base guanine plus PRPP to form GMP plus PPI. The hydrolysis of PPI drives the reaction in that direction. So if minute this reaction takes place, it will become irreversible because of the hydrolysis of PPI. So hypoxanthine to IMP, guanine to GMP is the reaction catalyzed by HGPRTase. Now the other reaction is adenine phosphoribosyl transferase, or what is called as APRTase. Now APRTase converts adenine, which is a base. You add sugar and phosphate to it from PRPP to form AMP plus PPI. So nothing, you write the names of the bases, you write PRPP and you get the involved nucleotides with it. So these are the two enzymes involved in the salvage pathway. What was salvaged here? The bases were salvaged here. Now there is a very important disorder associated with the lack of the salvage pathway enzyme. One of the enzyme is HGPRTase. If this enzyme is deficient, it can lead to a condition called as Leschnyhan syndrome. Leschnyhan syndrome is because of deficiency of HGPRTase. An MCQ question. So Leschnyhan syndrome, what is it? Let us look at it. It is an X-linked recessive disease. The deficient enzyme is HGPRTase. So if it is deficient, what will happen? It will lead to increase in the amount of hypoxanthine and guanine. HG stands for hypoxanthine and guanine. So increased hypoxanthine and guanine. So what will happen? They get degraded faster. Hypoxanthine gets degraded, guanine will degrade. The degradation product of purine nucleotides is uric acid. So it will lead to increased uric acid. When uric acid increases, it will lead also to increase in the PRPP, phosphoribosyl pyrophosphate. And this in turn will decrease the IMP and GMP. So the de novo synthesis is increased, more degradation, more uric acid. So it is a constant cycle that is happening out there. A lot of things are happening out there which will all lead to increase in the de novo synthesis. Now what is happening is, one thing is hypoxanthine and guanine. Remember, IMP, UMP are involved in the inhibition of PRPP synthase. So if inhibition is not taking place, at the same time the product is also less, the de novo synthesis is going to take place. Salvage is not occurring, de novo will occur. More nucleotides, more degradation, more uric acid. So it is a vicious cycle that is taking place in the Leschnyhan syndrome. So what will be the clinical features? One of the most important thing that is going to happen is hyperuricemia. Hyperuricemia, whenever uric acid increases, it is going to get excreted out through the kidneys, but more of uric acid in the urine will lead to uric acid stones, and that is nephrolithiasis, uric acid stones in the kidneys. At the same time, this uric acid can get deposited at different joints, and by forming monosodium urate. Monosodium urate crystals get deposited in different joints, leading to gouty arthritis. And in these people, remember, brain requires salvage pathway. So the brain gets affected. There will be motor dysfunction, cognitive deficits, behavioral disturbances. Now, one classic feature of Leschnyhan syndrome is self-mutilation. The person will be eager to cause mutilation. He will keep on eating himself, self-mutilation. Either the lips and all will be having a lot of hurt, and biting of the lips is taking place, biting of the nails, biting of different scratching, and he will try to use a knife or something and result in self-attack on him and cause self-mutilation. So this is a characteristic feature of Leschnyhan. So any case in which self-mutilation comes, you have to think that it is Leschnyhan. Leschnyhan means it is HGPR, that is the salvage pathway. Salvage of hypoxanthine and guanine is not taking place, and hence it can lead to brain and cognitive deficits and behavioral disturbances. Now, briefly, what is the role of PRPP in all this is phosphoribosyl pyrophosphate, MCQ, that's why I have told you. It is involved in de novo synthesis of purines and also pyrimidines. It is involved in the salvage pathway and also in the biosynthesis of nucleotide coenzymes like NAD, FAD, etc. So, in all these also PRP is involved. So, that is the role of PRPP in different de novo synthesis. Now let us look at till now we were all talking of when the ribose was ribonucleotides. Now let us talk about how is deoxyribose nucleotide synthesized within the body. So AMP, whatever was formed was ribonucleotide. How is deoxyribonucleotide? Why is it required? For DNA. Naturally, whenever DNA is undergoing replication, it requires deoxyribonucleotides. So the enzyme and its action, let me just show it to you. So AMP or ATP cannot be converted to the deoxy forms. It is always the diphosphate which can be converted to its deoxy form. If it is a monophosphate, it has to be formed diphosphate. If it is a triphosphate, also it has to form a diphosphate. Now the one I have given example is of ADP. The same thing holds true for GDP, CDP, UDP, etc. For all these, the deoxy mechanism whereby deoxyribonucleotides are synthesized is the same for all of them. They all have to be in the diphosphate forms. Now there is an enzyme known as ribonucleotide reductase. Ribonucleotide reductase. This enzyme is specific for the respective adenosine ribonucleotide reductase, guanosine ribonucleotide reductase, like that they are named as. So they are specific for the respective deoxyribonucleotides. So ADP gets converted to DADP and that will later form DATP or DAMP, however it is wanted. Now in this, as I said, ribonucleotide reductase is required. So this enzyme has got one more important component known as thioredoxin. Thioredoxin. Now thioredoxin is actually a peptide and it is acting like a coenzyme. And one important thing about thioredoxin is that it has a sulfhydryl group. So when it is giving its deoxy or it is converting ADP to deoxy, it gets converted to two molecules of thioredoxin in which there is a disulfide bond formation. So this gets reduced. When this gets getting like this deoxy, this is getting reduced. This sulfhydryl group is going to form a disulfide. So for the reconversion back to its active form, what is required? Whenever there is this like this, we require NADPH. NADPH again is converted to NADP plus and it converts it back to its active form. This enzyme is known as thioredoxin reductase. Now there is a role of selenium also in this thioredoxin and thioredoxin reductase. But right now let us remember that thioredoxin is the one which is responsible for the conversion of diphosphates to the deoxydiphosphates. Diphosphoribonucleotides to deoxydiphosporibonucleotides, and this is ribonucleotide. This is a ribonucleotide. How do you remember the names? You look at the substrate. This is a ribonucleotide reductase. I think the best way would be to write this as, it could be any base. Any base is converted to its respective nucleoside deoxy form. So this is the importance of ribonucleotide, and this is how they are synthesized within the body. So I have given it here, ribonucleotide reductase. This is specific for the diphosphates, that is important, and they form the respective deoxynucleotides. Thioredoxin is the peptide coenzyme, and this gets reduced. There is a formation of disulfide bond, and this is regenerated using NADPH. So I'll talk about the degradation of purine nucleotides. How is it taking place in my next session? Thank you.